A study about enzymatic laundry detergent by Grace, Jack, Margaret, and Sandra. First, let's talk about enzymes. One of the major industrial applications of enzymes is in the detergent area. So how do enzymes work? Enzymes are folded proteins, also known as a chain of amino acids, that act as catalysts in biochemical reactions, and are not consumed in the process, so therefore can be reused. There are many different sizes, shapes, and compositions of enzymes, however they all share a region called the active site. The enzymes used in the detergent industry are degradation enzymes in which the substrate is broken down to several components. Enzymes tend to be highly specific to certain molecules due to the specificity of their active site and as a result differ from normal chemical catalysts in this regard. Unsuitable pH or temperature conditions can affect the shape of the enzyme, therefore affecting the shape of the active site. This is called denaturing, in which the enzyme is no longer functional and nothing is able to bind to the active site anymore. Enzymes are catalysts and therefore lower the activation energy required for the chemical reaction to occur through binding of the substrate to the active site in the enzyme and the resulting orientation produced. Catalysts occur and resultant products are released. The induced fit model proposes that an enzyme can slightly change shape as to best fit the substrate based on their interactions. However, there is some debate over which model is a true representation of what occurs. The opposition to this model is the lock and key method. Now onto the types of enzymes. There are 5 types of enzymes used in detergents. The first and most common being proteases, which remove protein stains by degrading them into smaller molecules which can then be washed away. Examples include blood, grass, and sweat. Amylases remove starch residues by cutting starch into small sugars that then dissolve in wash water. Examples include potatoes, custard, and chocolates. Lipases degrade lipid molecules into small pieces which can then be emulsified. Examples include fats, butter, and sauce. Cellulase modifies the cellulose fiber structure by eliminating fuzz and pills on fabric so that the colors appear more bright and prevents microscopic dirt from getting into the fabric. Mayonnaise removes melanin stains by hydrolysis so that it can be washed away. But what are the required properties of an enzyme? There are 3 criteria that an enzyme must meet to be used in a detergent. Firstly, it must be stable and active at pHs 9 to 10.5. Secondly, it must be thermotolerant in the range from 55 to 100 degrees Celsius. And thirdly, it must be compatible with perborate, surfactants, chelators, etc. Now we will talk about bacillus. Alkaline serine proteases from Bacillus species make up more than 95% of the enzymes sold for laundry detergents. More than 20 different new proteases are fit to use in detergents, but most of the proteases that are used today are from the Bacillus species. This species of bacteria is used mainly due to its ability to secrete large amounts of extracellular enzymes. These proteases have broad pH profiles, from neutral to highly alkaline conditions, broad substrate specificity, and sufficient process and storage stability in the presence of surfactants and complexing agents. So why use enzymes? Enzymes have many desirable properties that make them a desired component of laundry detergent. Why use enzymes in washing powders? Enzymes allow dirt to be removed without the use of hot water, which allows for the washing of delicate or dyed clothes without the risk of damage, as there is usually at high temperatures. Therefore, money can be saved as less heat and energy is required. The reduction in chemicals transported to and from manufacturers also improves the carbon footprint. Difficult to remove stains such as blood or protein stains are able to be removed more efficiently with the use of enzymes. As enzymes are catalysts which speed up the reaction, they improve the efficiency of detergents. What are the effects of using detergents without enzymes? Clothes need to be washed at high temperatures, which can lead to damage to the fabrics. Detergent without enzymes is better for those with sensitive skin or skin conditions such as eczema, as enzymes are considered to be common household allergens. Enzymes that are not properly washed off clothes can cause a mild reaction when rubbed against skin. Non-enzymatic detergents can cause permanent stains when trying to remove protein stains. This is due to the oxidation and denaturation caused by bleaching and drying. Detergents without enzymes can make your clothes appear clean or stainless, but when clothes are ironed or placed in hot conditions, the stain melts and becomes visible again. This is because the detergent only removes the top layer of the stain, and the stain remains trapped in the fibre, whereas with enzymes the stain is removed completely. Now on to production.
So what goes into enzymatic laundry detergent? In general, the following are added to laundry detergent. Surfactants, which prevent surface tension between two substances. Builders, which are water softeners and remove ions in hard water. Optical brighteners that enhance colours and dyes. Bleachers, which target oxidizable organic stains. Antimicrobial agents that kill microorganisms. And enzymes, which are added in small portions, protease being the most popular. What are the requirements in enzymatic laundry detergent? Traditional chemicals compose the majority of a detergent's composition, with enzymes only being less than 1%. Other chemicals, sodium tripolyphosphate, have cleaning purposes, thus meaning biological detergents are not acting purely on enzyme action. Only a specific type of protein, serine, is allowed, as opposed to others. Thiol proteases are oxidized by bleaching agents, and metalloproteases complex with OH. Serine protease employs a catalytic triad, which is a group of 3 amino acids, aspartic acid, histidine, and serine, which are found in the active sites of some proteases involved in catalysis. Serine group contains an OH group that acts as a nucleophile. In addition, histidine is a proton acceptor from serine group, allowing this to occur. Nucleophile subsequently attacks carbonyl bond. Aspartic acid helps position histidine somehow, increasing its pKa and allowing histidine to act as a base. Aspartic acid deformation or mutation decreases the kcat by 10,000-fold. Serine protease allows for a covalent intermediate of the substrate and serine group. Ester hydrolysis allows the catalyst to reform. Now we will talk about the links between biocatalysis and green chemistry. Biocatalysis has many attractive features in the context of green chemistry and sustainable development. The first is that the catalyst, or enzymes, are derived from renewable resources and are biocompatible, sometimes even edible, biodegradable, and essentially non-hazardous. That is, it fulfills the criteria of sustainability remarkably well. The second feature is that biocatalysis avoids the use of and contamination of products, scarce precious metals. Some endangered elements, such as various noble metals, are used and is viably questionable, which raises questions in the cost of removing noble metal traces. A third feature is that reactions are performed in water under mild conditions, as biologically there tends to be ambient pH, temperature, and pressure conditions. Another feature is that enzymatic reactions have high activities and stereoselectivities and therefore remove the need for functional group activation, protection, and deprotection steps required in traditional organic syntheses. This means the processes are more step economic, energy efficient, and produce less waste. As a direct result of the higher selectivities and milder reaction conditions, biocatalytic processes often require more pure products than traditional chemical or chemocatalytic processes. Enzymatic processes, but not fermentations, can be conducted in standard multi-purpose batch reactors and therefore don't require any extra capital like high-pressure equipment. Biocatalytic reactions are conducted under roughly the same conditions of temperature and pressure, and hence it is relatively easy to integrate multiple reactions into eco-efficient catalytic cascade processes.